A very good day everyone. Today we will presenting you about the hydrogen production and the bioelectrochemical system, focused on the microbial fuel cell, and we will show you how we can address the clean energy crisis with an unexpected ally, which is the bacteria and their role in producing green hydrogen. We face a dual crisis. First is an urgent need for the clean energy and a booming demand for the hydrogen. Yet, over 95% of the hydrogen today is gray, which is made from the fossil fuel, creating a more carbon dioxide. This is the sustainability paradox we must solve. Why is hydrogen the future fuel? First, it's incredibly clean, emitting only water when used. Second, it has a high energy density, perfect for the heavy industry and transport. But the production methods are defective. Steam reforming from the natural gas is cheap but polluting. Electrolysis using the renewable power is clean but requires a massive amount of the electricity and is expensive. We need a smarter, more efficient way to make a green hydrogen. This is where the bioelectrochemical system offers a breakthrough. BES are devices that harness the metabolism of electroactive bacteria. Here's the key. This special microbe can breathe solid electrons. When they consume organic matter, like the wastewater pollutant, they release electrons. By capturing this electron flow in the circuit, we can generate electricity direct from the waste. This is the core principle of the most studied BES, the microbial fuel cell, or MFC. Let's look at the basic MFC. In the anode chamber, bacteria break down organic compound releasing electron and proton. The electron travel through a wire and creating a usable electric current, while the proton move through a membrane. In the cathode chamber, electron, proton, and oxygen combine to form a water. This result is the simultaneous wastewater treatment and a direct electricity generation. In conclusion, to meet the global demand for clean hydrogen, we must move beyond traditional, inefficient method. Bioelectrochemical system with microbial fuel cell provide a transformative solution. They leverage bacteria to turn waste into value, generating bioelectricity and offer a lower energy pathway to sustain green hydrogen production.
So now I will be explaining what microbial fuel cells or MFCs are. MFCs are a type of bioelectrochemical system that uses microorganisms under anaerobic conditions to convert chemical energy from organic matter into electrical energy. These MFCs have been used as one of the comprehensive solutions for three sustainability issues that are facing our world today, which are global warming, energy security, and wastewater management. Same as other fuel cells, these microbial fuel cells also have anode and cathode that is connected by an external circuit that helps in transporting electrons generated. And this is a full diagram of the microbial fuel cells. MFCs also consist of few main components that are working together to allow microorganisms to produce usable electrical energy. It consists of anode, cathode, membranes, electrodes, and circuit, as well as the microorganisms. First, anode here is the place where the oxidation will occur. The exoelectrogenic bacteria attach and release electrons. The microorganisms in the anode electrode oxidize the organic matter, breaking the pollutants and generating electrons that flow through an electrical circuit to the cathode compartment. It also consists of different materials such as carbon paper, graphite plate, carbon brush, and many more. At the cathode, oxygen accepts those electrons and reacts with the proton to form water. Between them we have the membrane, which works as a selective barrier. It only allows proton, which is H+, to pass through to the cathode side, while preventing oxygen from entering the anode compartment. This is very important to keep the anode oxygen free so bacteria can keep releasing electrons. So overall, the anode is the site of oxidation and electron release. The membrane only allows protons to pass through, and the cathode accepts the electrons to complete the reaction. Meanwhile, electrodes in circuit have a function to connect the anode and cathode in order to allow electrons to move and generate electricity. The microorganisms act as the power source and natural biocatalyst in the system. They break down the organic matter and release electrons and protons as part of their metabolism, Without them, no electrons would be produced and no electricity could be generated. And that's all for the key components of the microbial fuel cells.
Let's look at the mechanism of electricity and hydrogen. The first part is the microbial metabolism at the anode. Exoelectrogenic bacteria in the anode biofilm its organic waste and breathe the anode instead of oxygen. Simple organics like acetate is used for substrate oxidation. The first phase is the intracellular metabolism phase. This is where acetate enters the cell, is converted to acetyl-CoA, and oxidized via the TCA cycle. It releases electrons and protons. Electrons travel from center of the cell to inner cell membrane through inner membrane cytochromes. The next phase is the unique exoelectrogen phase, where bacteria utilizes three main strategies to bridge the gap between the outer membrane and the anode. The first method is to direct contact, where the outer membrane uses a C-type cytochrome that act like biological plugs. The second method is nanowires, where the nanowires can reach out and touch the anode, allowing electrons to flow over longer distances. And finally, we have electron shuttles. Chemicals act like shuttles, and they swim to the cell, drop an electron, swim to the anode, and drop a proton pathway. The electron pathway is where the bacteria at the anode exhales electron while breaking down waste. Electrons travel out of the liquid and into the conductive anode material. They flow through an external wire connected to a power supply to push the electrons to the cathode. They arrive at the cathode electrode waiting to react. For the proton pathway, protons are released into the water at the anode at the same time electrons are released. Protons stay inside the liquid, they migrate directly across to the cathode chamber, and they pass through a cation exchange membrane, which acts like a filter that lets protons through, but other chemicals don't. They arrive at the cathode to meet the electrons. Next is the hydrogen production mechanism. Bacteria oxidize organic matter at the anode, releasing electrons and proton. Electrons flow through the external circuit to the cathode. Protons diffuse through the solution to the cathode, and at the cathode surface, electrons and protons combine to form hydrogen gas. There are several differences between microbial fuel cells and microbial electrolysis cells. The first is the main output. MFC is electricity and the MEC is hydrogen. The reaction type for MFC is spontaneous, while the reaction type for MEC is non-spontaneous. The oxygen is required for MFC, but is not required for MEC. And MFC produces power, while MEC consumes small amounts of power. Understanding the mechanism helps us appreciate the different MFC designs and the substrates they can use.
Next, I'm going to explain the type of MFC and the substrate that is used for its operations. There are three common types of MFC, which is single-chambered MFC, double-chambered MFC, and stack MFC. Firstly, for single-chambered MFC, the anode and cathode are placed in the same chamber. It has porous cathode that expose directly to air, which utilize oxygens and allow proton diffusions. This design is simple and cost-effective. As for double-chambered MFC, it consists of two chambers where it's separated by proton exchange membranes or salt bridge. It has higher efficiency, however, the design is expensive. Lastly is stack MFC. Stack MFC is where multiple MFCs are stacked together to increase the total power output. Since one MFC only produces low voltage, stacking it up is required to reach the usable power levels. This allows scaling up MFC into real applications. Next is the substrate that is used for the MFC operation. What is substrate? Substrate is the important organic matter that will be oxidized by microorganisms in MFC and generating electrons and photons that involve in the productions of electricity. There are two types of substrate, which is simple substrate and complex substrates. Simple substrate is usually from single compound carbon source. It is pure and easily biodegradable, and the most common example for simple substrate is acetate. Acetate preferable because it is inert for the microbial conversion. As for complex substrates, it is the mixtures of more than one organic compounds, for example carbohydrates, proteins, fats, and other molecules. Usually, it's sourced from wastewater such as domestic wastewater, municipal wastewater, and the sewage sludge.
Next, let us learn about the application and limitation in MFC. First, what is the real-world application of MFC and how are they used in this application? Microbial fuel cells or MFC have gained attention because they can treat wastewater while generating small amount of electricity. They have been applied to domestic, industrial, and agricultural wastewater, including brewery waste and palm oil mill effluent. For example, palm oil mill effluent treatment in Malaysia can cause
strong COD reduction because MFC can remove COD, nitrogen compounds, and even heavy metal. Another important application is biosensor, especially for detecting water quality. MFC-based BOD sensor provide rapid water quality measurement, which is useful for industry that must monitor their effluent. For example, when pollutants like heavy metals enter the water, the bacterial activity drops and we can see an immediate drop in voltage. In addition, this system supports bioremediation by degrading dyes, pesticides, and even reducing heavy metals. In contaminated soil and sediment, microbes naturally break down pollutants. For example, textile dye wastewater in India can be decolorized with MFC reactors. Now I will explain the main limitation of microbial fuel cells. First is a low efficiency and power output. MFCs still generate very small amount of electricity because of high electrode reaction and limited electron transfer by the microbe. This means that they are not suitable for large scale or high power application like industrial energy production. The second limitation is the environmental sensitivity because the changes in pH, temperature fluctuation, toxic chemical or even variation in the substrate can reduce their performance. This makes real-world operation less predictable and harder to maintain. The last limitation is the high cost of material and technology. Components such as electrodes, membrane, and catalysts are still expensive to this day. Researchers are exploring cheaper alternatives like agro-waste material, but they are not yet proven for commercial use, which slow down the adoption. Even with limitation, the future of MFC is still promising. Here is what the next generation of hydrogen-producing MFC could look like.
As we've seen, microbial fuel cells already offer an exciting way to produce clean energy from organic waste. But what makes this technology even more promising is how quickly it's evolving. One major area of progress is the development of advanced electrode materials. Traditional electrodes are expensive and not very efficient, but newer options like carbon cloth, carbon nanotubes, and graphene are showing huge improvements in electrode transfer. These materials could make MFCs cheaper, more durable, and more productive. Another important direction is genetic engineering of microbes. Scientists are now creating bacteria that can transfer electrons faster, tolerate harsher conditions, and break down more complex types of waste. This means future MFCs could generate hydrogen more efficient and operate in a wider range of environments. Membrane technology is also being redesigned. New proton exchange membranes aim to reduce cost, prevent fouling, and speed up proton movement, all of which directly increase hydrogen output. And finally, we are seeing rapid growth in industrial integration. Wastewater treatment plants, food processing factories, and agricultural farms are starting to test MFC systems to turn their waste into usable energy or hydrogen. If scaled successfully, MFCs could become a key part of future smart cities focused on circular, zero-waste energy systems. In conclusion, microbial fuel cells offer something rare, a technology that can clean waste, produce electricity, and generate hydrogen all at the same time. Although challenges like low power output and material costs still exist, the combination of better materials, engineered microbes, and industrial integration shows that MFCs are moving from experimental research to real-world application. By turning what we throw away into clean, renewable energy, MFCs represent a powerful step towards a sustainable future.
This has been our presentation on hydrogen production and bioelectrochemical systems. We hope you enjoyed learning about how bacteria can power the future. Thank you!